Welcome to the Mind and Matter podcast. I'm your host, Nick Jikomes, and today I'm speaking with Dr. Tommaso Di Ianni. Tommaso is an assistant professor in the departments of Psychiatry and Behavioral Sciences and Radiology and Biomedical Imaging at the University of California, San Francisco, and his lab is studying neuroscience. They use really interesting technology like ultrasound to modulate what the brain is doing and to actually image what's going on in the brain. So we talked about how some of that technology works in the context of experiments they're doing in rodents to look at how ketamine is actually working in the brain and what it's doing. So ultrasound, it's the same technology that a pregnant woman is exposed to when she goes in and gets those images, those first images of her baby. So they actually use ultrasound to manipulate the brain and to see what's going on in the brain. And so we talked about some of the experiments they did using this technology in rats, looking at how ketamine influences different parts of the brain. So they're actually able to give ketamine to rats and obtain information about how proxies for brain activity are responding to ketamine. So they look at different parts of the brain, from the prefrontal cortex to the nucleus accumbens and other regions. They look at how patterns of activity, in essence, are responding to ketamine, and they unveiled some interesting results. So they found that there are sex-dependent effects of ketamine in the brain. In other words, male rats, the patterns that they see in their brain are different than what they see in female rats. They also uncovered some interesting results with respect to the opioid system. So some of ketamine's effects in the brain seem to involve the opioid system, and we talked about why that is and what they saw. And interestingly, the involvement of the opioid system in some of ketamine's effects is itself sex dependent. So they see different levels of involvement of the opioid system in males versus females. And so we talked about some of that recent work that they've done and how that fits into the larger field of ketamine research. We talked about what is known and still unknown about ketamine's mechanisms of action in the brain. We talked about some of the controversies in the field with respect to, the extent to which ketamine is addictive or less addictive than other drugs, how exactly it's working in the brain to elicit its antidepressant effects, and how Tommaso's recent results tie into all of that. So if you're interested in ketamine and the brain, how it's working, and what's going on in terms of the latest findings, this is a really good episode. As always, don't forget to check out the links in the episode description. From there, you can get to the Mind and Matter Substack at mindandmatter.substack.com to see all of my long-form science writing, which integrates and synthesizes a lot of the information across different episodes of the podcast. My free weekly newsletter is also there that tells you upcoming guests, new episodes, interesting research that I'm reading that informs who comes on the show, and other things that I'm looking at. You can also help the podcast by clicking on some of my affiliates. So I link to 2 or 3 different companies that I think make really cool products, from at-home blood testing products to metabolic tracking, technology, to nutrition. These are things that I have and I use myself. If you click those links, you will get a discount for those products, and that helps the podcast a little bit. I get a little bit of that money, which I use to invest in new technology and to travel to guests. I'm going to start doing that more, to do in-person interviews and things like that. And your support there really helps. And with that, here's my conversation with Dr. Tommaso Di Ianni.
Reaching out. I was actually listening to your podcast quite a bit when I was commuting to Stanford. So that was
which ones did you listen to?
Oh, I listened to Alex Kwan. I listened to Gul Dolan more recently. I listened to Christian Luscher.
Oh yes. I'm maybe going to ask you about some of that work and how it connects.
Yeah. I listened to quite a few.
Excellent. Yeah, no, I'm glad to hear it. Sometimes I'm starting to hear that more, which is a nice surprise.
Yeah, that's nice.
Yeah, because when I first started, I was essentially emailing people and they have no idea who I am. But sometimes now they're like, oh yes, I listened to this episode, so I already know you.
Nice. Yeah.
Do you want to just start off by introducing yourself, telling people a little bit about your scientific background and what your lab is doing now?
Sure. Yeah. So I am Tommaso Di Ianni. I'm an electrical and biomedical engineer by training. And then I did a postdoc at Stanford. I was in the Department of Radiology, but I was really working at the interface of ultrasound and neuroscience. So my background is mostly in ultrasound, so both ultrasound imaging and therapeutic ultrasound. And during my postdoc at Stanford, I started applying these modalities to neuroscience, and more recently, we started both developing and applying developing technological innovations and applying this modality called functional ultrasound imaging to studying pharmacology and studying how drugs act on the brain, and more specifically ketamine, which is what we're going to talk about today.
Hmm. Yeah, let's start off by talking about some of the technology here. So you're using ultrasound to do imaging. Normally, when people hear ultrasound, they think about a pregnant woman going in for her ultrasound to get those first images of the baby. Can you give us an overview of how exactly ultrasound technology works and what some of these other applications are beyond the familiar one?
Yeah. So the imaging is really not much different than what happens one of the applications, the one you mentioned, for fetal imaging. But what we are really looking at here is blood flows. So ultrasound is also imaged. So people may also be familiar with applications like color flow mapping, like when you're imaging an organ like the liver, for example, or the neonatal brain, or the arteries, for example, in the neck, and you see those blue and red colors overlaid on the image. Those colors show a measure of blood flows. And so in the application that we are using, that is again called functional ultrasound imaging, we are the underlying technique is called power Doppler. So we are doing Doppler imaging and imaging blood flows. Now, if we have one single image of the blood vasculature, or like the blood flow, that's just giving us a snapshot of the vasculature at that specific time point. But what we do here, we track those blood flows over time, and there is a principle the principle that is underlying all this is called neurovascular coupling, which is telling us that essentially the brain has no intrinsic ways to store energy and oxygen, and therefore, if there is an increase in activity locally, the way these cells can get the supply of oxygen and glucose that they need is by recruiting more blood. So if we track those blood flows, we can indirectly infer neural activity. So this is not dissimilar to what happens with other modalities like fMRI, for example, functional magnetic resonance imaging.
Yeah, so a chunk of neural tissue becomes more active. It could be excitatory cells, it could be inhibitory cells, it could be both, but they're becoming more active. They require more energy to sustain that activity. So blood flow increases to that region to supply them with oxygen and nutrients and this and that. So it's similar to fMRI in that way.
In that sense, yes. We are looking in it's similar to fMRI in the way that we are looking at blood flows as an indirect measure of neural activity.
Yeah.
fMRI more specifically looks at the rate. It looks at oxygenated and deoxygenated hemoglobin, while in this case we're looking at cerebral blood volume. But the two are highly correlated, and indeed people have looked at both functional ultrasound imaging and fMRI back to back with optogenetic stimuli, for example. So like stimulating neural activity in a very well-controlled way, and imaging using the two modalities. And what they saw is that functional ultrasound is indeed very highly correlated to fMRI. So they are giving us pretty much the same information, but functional ultrasound. The nice thing of functional ultrasound imaging is that it is much more sensitive than fMRI. So for studying these kind of like neural activity and how, for example, how the brain reacts or how the brain responds to drugs in animals, functional ultrasound imaging gives you kind of a better resolution and better effect sizes.
Okay, so just for people listening, when we look at this data and we talk about it, you guys are measuring a proxy for neural activity, which has to do with blood flow, but you're not directly measuring the action potentials that the cells are sending out.
That is correct. Yes. And so, yes, exactly. There is that caveat that we are not measuring spiking activity. but it's somewhere in between. So I like to
Hmm.
think of it as kind of a translational tool in that whatever we find in animals, we can possibly translate into humans with fMRI, for example, or we can look at kind of correlative measures in humans with fMRI because it's the underlying mechanism is very similar. So it's I think it's a nice bridge between the animal world and the human world.
Hmm. And you said something earlier about Doppler imaging. Does this have to do with the Doppler effect?
Sort of, yes. I mean, the idea, so kind of in the kind of at the origin of ultrasound blood flow imaging, there is the Doppler effect, and that's because kind of so for people listening, the Doppler effect is essentially that effect when kind of an ambulance is approaching, and then you hear the pitch is increasing, and then when the ambulance is kind of going away from you, you hear a lower pitch. So you have that, but the ambulance is emitting the same tone
Hmm.
all along. So that modulation that you perceive is called the Doppler effect, which is essentially squeezing the pressure waves when the ambulance is approaching. So you hear a higher pitch because the waves are closer together. And then when the ambulance is leaving, is dilating those pressure waves, and so you hear a lower pitch. Similarly, when you send an ultrasound pulse and you have a scatterer moving away from you, it will modulate the ultrasound that you receive. And if it's moving towards you, so you will have either a higher or lower frequency, and then
Hmm.
therefore by looking at those frequencies, you can you can kind of infer the velocity of the scatterer.
Yeah, and let's just be really explicit for people. So when you use an ultrasound device, how exactly is it working to ultimately generate the data that we're going to talk about?
So there is a lot happening in there. So I'm trying to kind of squeezing in a lot, as much information without as possible without making it too confusing. So we are sending ultrasound pulses, and then so every time we send the pulse, then we start listening.
Hmm.
So as
And
that pulse,
it's ultrasound.
it's
So
ultrasound.
are you talking about a
Yes,
physical vibration that's generating a
it's
sound
a
wave?
exactly. It's a pressure wave that is not dissimilar to sound. The only
Hmm.
difference is that so we define ultrasound whatever is beyond 20 kilohertz because the spectrum of the kind of the audible sound in humans is between 20 hertz and 20 kilohertz.
Mm.
Um, Almost nobody can get to 20 kilohertz, but that's
Yeah,
kind of
yeah.
like the textbook
I
definition
see.
of the
So
audible
ultrasound
spectrum
is basically just referring to sound waves beyond what humans
beyond
can
exactly.
possibly perceive.
Exactly. And so in our case, we're talking about megahertz here. So the higher the frequency, the better the resolution. So we want to use a frequency as high as possible because then we get a better kind of a smaller pixel, kind of a higher spatial resolution. Um, but then, so essentially, yeah, we send up an ultrasound pulse, we listen, and then we use some smart array signal processing. So in the ultrasound probe, that is the one that is in contact with the body, in contact with the brain in our case, or in contact with the kind of with the skin in all the other clinical applications. That's what we call an ultrasound probe. And the probe is nowadays it's almost never made of one single ultrasound transducer. So it's really made of an array of transducers. And so in the back end, so what happens in the scanner, we are detecting the pressure waves that are backscattered, that are returned, like scattered back by the tissue
Hmm.
from an array of sensors. And then we use some signal processing to essentially refocus artificially, if you will,
Hmm.
so create electronic lenses.
I see.
So
So
we refocus in all the
yeah.
pixels in our image.
So it sounds like there's an analogy between optical forms of imaging. So instead of using photons that go in and come back and they scatter with some kind of pattern, you're just doing this with sound
To some extent, yeah. I
to
mean, I
some
guess
extent.
you could think about it that way. somewhat, yeah. I mean, it is not. We're delivering some form of energy inside. In this case, is ultrasound. In the other case, it would be optical energy, and then you have something that it's yeah. It has to be scattered in this case, right? Because we are staying outside of the body, so kind of we are sending something in and then listening or watching from the same location. So there is a backscattered component.
Hmm.
Um, so yes, I mean it's not dissimilar. The signal processing also kind of the array signal processing that I was talking about really comes from radars, and it's not like in radars there are these kind of antennas that are called phase arrays that are essentially moving the electromagnetic beam to scan a volume. And in this case, we do something similar as well. So by having access to all those tiny ultrasound elements, we can manipulate the ultrasound that we send in because we have access and we can control each of those elements electronically. So everything happens electronically. We're not moving anything mechanically, but then we can um, manipulate the ultrasound very well and reconstruct the images out of the ultrasound signals that we receive from
Hmm.
all these tiny little transducers.
And last question on this tech. What kind of can you say a little bit more about the spatial and temporal resolution that you're actually getting?
Yes. So as I mentioned, the spatial resolution in particular is a function of the frequency. So the higher the frequency, the higher the spatial resolution. However, the attenuation is also a function of the frequency. So the higher the frequency, the higher attenuation we will have to face. And so for applications that are relatively shallow, we can use much higher frequencies because we can still penetrate a few centimeters without losing too much of our kind of signal to noise ratio, without losing too much energy. And so, for example, in our case, we're using 15 megahertz in all our functional ultrasound imaging studies. And then applications, for example, like in the liver, where you're imaging down to 15, 20 centimeters, those applications use much lower frequencies. And so that's in kind of as the flip side of the coin is that you can penetrate deeper, but then you get a lower spatial resolution.
I see.
Just yeah, one caveat actually that I wanted to mention, since we're talking about penetration depth compared to optics, is that the nice thing of ultrasound is that we can penetrate deep. So we can, in our case, for example, we can image the whole body
Yeah, yeah. So
without
for
having
those
to
listening,
access.
so when you use a microscope to do like calcium imaging or something, you can image the cerebral cortex. You can go down, a little bit in the brain and collect photons back to create an image that way. So what you're saying is you can go much deeper in the brain and get information about deeper structures.
Exactly. And possibly we can get whole brain imaging. Or like, going back to optics, the other thing that you could do, you can implant, for example, a fiber optic, the
Hmm.
technique that is called fiber photometry.
Hmm.
And so you can detect photons from deep inside the brain, but then you are so
You're
one
also causing physical damage, though. So
exactly, it's
yeah, yeah.
invasive, and also you are biasing in the study in that you are deciding a priori where you want to look at.
Mm.
While when you look at the whole brain, you
You can survey the whole thing and then decide.
exactly,
Yeah, yeah.
exactly.
Last question, actually, one more. Is there what is there any reason to believe that the ultrasound stimulus itself might affect neural activity?
That's a very good question. So we ourselves, like in our lab, we are we're working with ultrasound in neuroscience. So we work both on we develop technologies and we use ultrasound imaging as well as another technique that is called ultrasound neuromodulation. So we are using ultrasound to modulate neural activity. So we are actively kind of doing that as well. And in that case, kind of so the difference is that we need a higher level of intensity or a higher energy at the at the focus or at wherever we want to, in that case, stimulate or inhibit neural activity. it requires much higher intensity than what we use in imaging. Because in imaging, so to give you an idea, in one case, we are focusing the energy. So we are using those elements or a physical or an electronic lens to focus the ultrasound energy in one spot, even though the intensity is still much lower than what is called high intensity focused ultrasound, which is essentially ablating tissue. So we're not in that regimen of very high intensities, but still the intensity that we deliver for neuromodulation is a lot higher than what the intensity that we subject the tissue to in ultrasound imaging applications. Because in ultrasound imaging, we are using plane waves, and actually that goes back to the temporal resolution question, which I haven't really answered yet. But we use plane waves, which means we are insonifying. We are sending ultrasound into the whole field of view at once. So we're not focusing the energy in any point. The energy is diluting in the tissue, is being diluted in the tissue. So the intensity at the kind of in the tissue is very minimal compared to what we would need to modulate neural activity.
I see. So, okay. So it's in that way, it's different from something like two photon microscopy, where you're focusing all of your light in one plane. Yeah,
exactly. So in the old days of ultrasound imaging, that's what happened. So you would focus the energy in one point and then either build kind of build up, meaning like in post-processing, right? I mean, you acquire the data and then you build the image in that specific point where you focus the energy, or you build a line. So you have a focus, because you have focusing in transmit and receive. So kind of you can do all these different tricks. So basically you can, anyway, I mean, what I was trying to say is that in the old days of ultrasound imaging, or in most clinical scanners, still today, you are focusing the energy in one specific point or in a few points for each image line, okay? While in our case, we are sending the energy in the entire volume at once. And the advantage of that is that every time we send a pulse, instead of sending energy in one point and therefore imaging that one point or a line, we can image the whole volume. So we have a much higher temporal resolution in the order of kilohertz. While if we have to build an image point by point, by the time we have built the whole image, we are already, like, the tissue has moved, everything has changed. So that kind of gives us a much lower temporal resolution that we would actually need to look at these tiny signals from the brain musculature.
So using ultrasound, you can get information about what's going on in the brain. You're getting a signal back, which has to do with blood flow, which is going to be related to what the neurons are doing in terms of their action potentials, but it's not literally a direct measure of that signal. It's kind of similar to fMRI in that way, but we're using ultrasound, the same thing that a pregnant woman would, when she goes in to get her ultrasound to see her baby. It's the same type of technology. You can point that at a little rodent's brain and actually get information about pretty much all parts of the brain. And you guys used this technology recently to study the effects of ketamine in the brain. Because I've done so many episodes about ketamine, and enough people listening will have some basic education here, I don't want to spend too much time on the basics of ketamine. Nonetheless, let's just, for those listening who aren't very up to speed on what's going on in the ketamine research world, what is ketamine? What has it been used for historically? And what have we started to use it for more recently from a medical and therapeutic perspective?
Yes. So ketamine, the textbook definition of ketamine is an NMDA receptor antagonist, which means it is blocking these receptors in the brain that are called N-methyl D-aspartate receptors. And historically, ketamine was really used as a combat anesthetic in the beginning. So it is very safe compared to other anesthetics, compared to narcotics, for example. It doesn't cause significant side effects, so it can be used on the battlefield. And so that's how kind of ketamine was used for historically for a very long time. And then in in the early 2000s, it became clear, or there started to be evidence that ketamine had antidepressant effects, and at very low doses. So at a very subanesthetic doses, so a very tiny infusion of ketamine has antidepressant effects. And the nice thing is that those effects are very rapid. So it's a rapid acting antidepressant, which is an advantage compared to, let's say, um, selective serotonin reuptake inhibitors that take weeks to show effects. So that's one advantage. However, the antidepressant effects are also short-lasted. So
Hmm.
it starts acting within hours, and then usually the effect fades out between 1 and 2 weeks.
I see. So, immediate effect, rapid antidepressant effect, but not particularly long lasting compared to other drugs.
Correct.
Even things like psilocybin, for example, have a much different time course. You mentioned that you get different effects at different doses. So, just to summarize that, at high doses, let's just call it high doses, you get anesthetic effects. That's been a historical use. At lower doses, you get psychoactive effects. So when people are using ketamine recreationally, I guess we can just call that a medium dose.
Yeah.
And then the low doses that give the antidepressant effects, are they low enough where there's no obvious psychoactive effects either?
No, there are definitely psychoactive
Yeah.
effects.
I mean,
It is
beyond, I
it is
guess the anti,
dissociative,
yeah. Okay.
mostly.
So the antidepressant effects do happen concurrently with some dissociation.
Um, yeah, I mean the dissociation happens. So I believe the antidepressant effect outlasts the dissociation. So the
Mhm.
dissociation is only limited to the half-life of the drug,
I see.
kind of the time when the drug is actually actively uh, inside the body, and then the antidepressant effects outlast that, meaning that, as I mentioned, they last for 1 to 2 weeks, and at that point, ketamine will be
Mm.
definitely out of the body, so it's not actively
Yeah.
causing any. It's not binding to those receptors
Yeah.
anymore because it has washed out,
Yeah. The
and
antidepressant
but there
effects
is
are
still an antidepressant effect.
yeah, the antidepressant effects are triggered by the drug, but they outlast the drug. So something's
Correct.
happening that outlasts the drug being in the brain. That actually sort of preempts my next question. So before we go into your work, which is going to have interesting things to say about how ketamine's working, you mentioned that the primary mechanism that ketamine is known for is antagonizing the NMDA receptor in the brain. Has that been proven to be the mechanism that causes or is required for the antidepressant effects? Are other mechanisms thought to be at work, that are actually responsible for the antidepressant effects or that are also required in addition to the NMDA receptor effect? What's sort of the latest on what's known and what's controversial about the antidepressant mechanism?
I think that's a very active question right now, because there is no. I don't think there is definitely not necessarily a consensus on the mechanism, definitely in humans. Um, because again, the original idea is that ketamine binds to these NMDA receptors, preferentially in inhibitory interneurons in the prefrontal cortex, and therefore it is inhibiting inhibitory interneurons and having an excitatory effect, kind of a net excitatory effect. But there's been evidence recently, preclinical evidence showing that there are also other mechanisms that may be responsible for the mechanism of action of ketamine, at least for the kind of the antidepressant-like effects in rodents, for example, on AMPA receptors. So it's another receptor that also is a glutamate receptor, but different than the NMDA receptors. And then more recently, both in humans and in rodents, there's been evidence that by blocking the opioid receptors, so a different type of receptors, um, that seems to modulate the antidepressant effect as well. So now how those are kind of placed in the pipeline, so where ketamine is binding first and what is happening downstream, at least to my knowledge, that is not very clear yet. So we
Hmm.
don't know exactly what's the cascade of events that is leading to the antidepressant effect downstream.
I see. Another way to start to think about this is, there are many other drugs that antagonize the NMDA receptor. Some of them are probably much more specific for interacting with the NMDA
Correct.
receptor. Do NMDA receptors antagonists generally have antidepressant effects, or is ketamine somehow unique here?
No, that's one of the exactly, yeah. Thank you for that question. One of the reason why I believe people have started in the first place have started investigating other mechanisms for the antidepressant effect of ketamine is that more selective NMDA receptor antagonists didn't work very well in clinical trials. So they would still work maybe in animal models, but the animal models are models by definition. So kind of they don't, we can only model a very complex disorder like depression in animals, we can only do that much to model that. So because it's a very complex disorder, and therefore, anyway, those NMDA receptor antagonists didn't work very well in clinical trials, and therefore people started looking at alternative hypotheses for the mechanism of action of
Mhm.
ketamine.
Um, is there any evidence that ketamine might be metabolized and one or more of its metabolites might be doing something relevant?
Therefore, so definitely, so ketamine is metabolized into norketamine, hydroxynorketamine. So there are all these um, metabolites that are um, created, if you will, mostly by the liver, and then they're also psychoactive, so they reach the brain and they do something themselves. There is some evidence that those metabolites are actually sufficient to create um, an antidepressant effect in
Hmm.
animals, but there is also evidence that is going against that. So I don't think we have a clear picture yet if specifically hydroxynorketamine, if it is, at least to my knowledge, um, if that is sufficient to have an antidepressant effect.
Yeah. So this is a very active field of research. It sounds like there's evidence for some of these things, but then there's also conflicting evidence. So we really are on the cutting edge here, and no one truly knows exactly what's going on yet.
That's correct. Yes, I think that's yeah, we can.
Um, One more background piece of information about ketamine that I think is interesting and relevant. So, as many people know, there's two ketamine isomers, S and R. What do we know about the differences between them? Do they have different pharmacological properties? Do they have different effects with respect to the antidepressant effect or anything else?
Yes, definitely. So first off, maybe let's step back. So we have three compounds here. Right? We have really like three. When we talk about ketamine, we are talking really about three different things. One is S-ketamine and R-ketamine. So these are the two stereoisomers. So it's the same molecule but specular structure. And then the other one is racemic ketamine. So usually when we talk about ketamine without specifying anything, we are talking about the racemic compound, which is more or less 50% R-ketamine and 50% S-ketamine. And what for what we know, at least now, I think yes, they are all acting differently, and they have different um, affinity for different receptors. S-ketamine, for example, seems to have a higher affinity for opioid receptors or for mu and kappa opioid receptors than R-ketamine, for example. Um, so they are definitely they act differently, and um, S-ketamine is FDA approved for treatment-resistant depression. R-ketamine, it's an active field of research to determine if R-ketamine has antidepressant effects or not. And the safety profile also seems to be different between the two, at least in animals.
Mm.
So they are definitely different molecules and they act differently. Yes.
In the study that you guys did recently that we're going to talk about, did you use one isomer or was it the racemic ketamine?
Um, So we worked with both. So the study that you are mentioning, the one that was um, published more recently, that's with only with racemic ketamine. And but we also collaborated with Mike Michaelides at the National Institute on Drug Abuse at the NIH. And in that case, we used S-ketamine. So we were imaging the effects of S-ketamine in the brain. And in the paper that came out recently, we were only looking at racemic ketamine. So it's, again, 50% of R-ketamine and 50% of S-ketamine.
Yeah, so let's talk about the recent study a little bit. Can you just give us a basic overview? What was the setup and what kind of what kind of questions were you looking to answer?
Yes. So our study really kind of the way we came up with this idea of testing the opioid receptors, testing kind of what is happening in the brain at the opioid receptor level, um, was generated by a couple of papers that came up in between 2018 and 2019 that were showing then that in patients, so patients with treatment-resistant depression, um, an opioid receptor antagonist called naltrexone was suppressing the antidepressant effect. So in these patients, they were pre-treating the patient with naltrexone before administering subanesthetic ketamine. And in the other group, they were just administering ketamine. So they were pre-treating with saline, it's kind of an inert compound. And in the group that they pre-treated with saline, so the group that just received ketamine, they saw, as expected, an antidepressant effect of subanesthetic ketamine. However, when they pre-treated the patients with naltrexone, they were suppressing that effect. So they were essentially deleting, at least partly, the antidepressant effect of ketamine.
So the interpretation of that is that somehow the opioid system is involved.
Yes, that's correct.
Okay, so what did you guys do in your paper with rodents?
So in our rodents, in our paper, we essentially tried to back translate, kind of reverse translate that into animals. Now, of course, we don't have depressed animals. Just as a caveat, these are rats, and so it's not a tiny human. So it's a different animal altogether. But we tried to kind of reverse translate that study. So we use the same compound that is naltrexone again to block the opioid receptors, and then we administered subanesthetic ketamine at a dose that is widely used in the literature and is known to elicit antidepressant effects or antidepressant-like effects in rodents. And then we imaged essentially what happens in the brain when we administer ketamine alone or when we are blocking the opioid receptors before we administer ketamine. And so, and the underlying idea is that if ketamine is not interacting with opioid receptors at all, then the two conditions should look
The same.
the same.
Mm-hmm.
But if a ketamine or if the opioid receptors are responsible to some extent that we don't know yet, if they are responsible for that effect, then when we block the opioid receptors, so when we take them essentially out of the picture, we silence them, then there is something different happening. And that's what we try to decode here.
I see. So the question is basically, is the opioid system involved in ketamine's action in the brain? You have this ultrasound technique. You can point your little ultrasound at the skull of a rat. You can get information about what's going on in the brain. I'm going to refer, just for people listening, to connect this with what we were saying earlier about how this technique works. I'm going to say things like brain activity, but you're not measuring spiking activity. You're measuring a proxy for this, but it's related to to how active the neurons are. So you're able to look at activity in different parts of the brain using this ultrasound technique. You can give ketamine and simultaneously block the opioid receptor. If that receptor system, if opioids are doing something, then we should see a difference between ketamine versus ketamine plus opioid block. So where did you look in the brain? And then what did you see? Did you see a difference?
So we looked at we limited our, because we didn't have a fully volumetric imaging setup at the time. We looked at two slices in the brain, but we tried to pick slices where we knew from the literature that something interesting could happen. So in one slice we have the prefrontal cortex that is been has been shown to be involved with the mechanism of action of ketamine broadly for different models of disease, and also more mechanistically, the prefrontal cortex seems to be very heavily involved with the mechanism of action of
I think
ketamine.
it's fair to say, right, that like certain circuits in the prefrontal cortex, certain chunks of the brain in that region seem to play an outsized role in depression, generally speaking, and in the action of things like ketamine and serotonergic psychedelics.
That's correct. Yes. I mean, it seems that exactly. But honestly, all those regions that we are going to talk about, they're both interesting for both depression and ketamine, and so kind of that was interesting for us to see that was actually the case. So the other region in that same slice is the nucleus accumbens, which is a region that is very important for reward processing. So also important for depression, for example, for the anhedonic aspects of depression, so kind of lack of pleasure from rewarding stimuli. So the nucleus accumbens also, again, like a very, very important region.
Yeah. And that's also like when we think about things like drug addiction or addiction generally, that's a kind of alteration of reward processing. And the nucleus accumbens is a very important part of the brain. Circuits in there are very important for addiction. Before we go further in your paper, actually, can you give people a short summary of what do we know about ketamine's addictive potential relative to other drugs of abuse?
Well, just for full disclosure, I'm not a psychiatrist, but I will just, so I don't see patients with either depression or addiction, but I think in general, the idea is that ketamine is not, or the kind of the mainstream idea is that ketamine is not very addictive. It is abused, so it is a substance that it is a controlled substance, so it's regulated by the Drug Enforcement Administration, and the reason is that it is used recreationally, it is abused as a drug. But kind of the general consensus, at least to my understanding, is that it's not very addictive. But again, this is something that is very much an object of research right now. And there, again, seems to be a difference also between the different molecules, like R-ketamine versus S-ketamine versus racemic ketamine versus hydroxynorketamine and so on. So we don't know exactly what is happening, and we don't know exactly if it is addictive or not.
Yeah, I mean, we don't have the mechanism of action fully worked out. That's part of what your paper's about. You've got R-ketamine, you've got S-ketamine. You know, sort of my take on this, just for those listening, is on the one hand, you've got animal research from people like Christian Luescher, who I've had on the podcast before, if you want to go learn about that. And what they show fairly compellingly is that ketamine is certainly less addictive in a rodent than something like cocaine. That doesn't mean it's not addictive at all. And it's also important to keep in mind, right, even though it's obvious, it's easy to forget a lot of times that, you know, rats and mice aren't humans. Because even though that research in animals shows that there seems to be quite a low addiction liability for ketamine, if you've spent any time around people who take ketamine recreationally, there are people out there who get addicted to ketamine for sure. So we kind of, yeah, we don't know exactly how addictive yet, but it's probably less addictive than something like cocaine, which is quite addictive.
That's correct. But then on that note, also work by Mike Michaelidis, our collaborator at NIDA. NIDA is the National Institute on Drug Abuse. They have looked at R-ketamine versus S-ketamine, and also in the other paper that I mentioned, where we looked at functional ultrasound imaging with S-ketamine in the nucleus accumbens again. They show that rats are self-administering S-ketamine, which means that they are kind of self-administration is one of the is a behavioral model that is taught to recreate some of the aspects of addiction in rodents, and rats do self-administer S-ketamine, at least in their hands, at least in their papers.
I see. So there could be a difference between S and R, and there's some
Exactly.
evidence that S is reinforcing in rodents, which would imply some addictive potential in humans.
Exactly. And racemic ketamine again is made of both, so we also have that S-ketamine component.
Okay, so you looked at nucleus accumbens in your paper. You looked at parts of the frontal cortex. What did you guys see?
Well, we also looked at a more posterior slice where we have some other interesting regions, like the lateral habenula, for example, which is a region that has been implicated heavily both with ketamine and with opioid receptors. It is also a region that is part of the reward circuit. It's not been studied a lot, definitely less than the prefrontal cortex and the nucleus accumbens, but what we know is that it is also implicated with the reward circuitry in the brain. And in that slice, we also had the retrosplenial cortex, which is also another region that has been recently shown to be involved with the mechanism of action of subanesthetic ketamine, and specifically for the dissociative effects of subanesthetic ketamine.
Interesting. All right, so you looked at several brain regions using this technique. We've got the ketamine condition. We've got the ketamine plus blocking the opioid receptor condition. What was the basic result that you saw?
So essentially, we saw that when we are pretreating the animals with opioid receptors, it is modulating the neural activity evoked by subanesthetic ketamine. So in some regions, it is suppressing, at least partially, that neural activation. In some other regions, it is enhancing the response to ketamine. So it appears to have divergent effects in different regions, which is also somewhat expected because some of these regions are acting differently, for example, in that reward circuit and in other circuits as well. So that was expected, but there seems to be definitely an effect that is mediated by opioid receptors, because when we take them out of the picture, something is changing in the brain. However, something that was very, very surprising and unexpected to us was that this was only the case for male rats. When we did these experiments in female rats, we did not see essentially any differences, at least in these two slices that we looked at. So there seems to be a sex divergence in kind of this opioid-mediated component in the response to subanesthetic ketamine.
What exactly was the difference between males and females?
Well, in males, we saw all these different regions showing statistically significant effects. In females, we almost did not see anything. So we saw some minor clusters of differences, but they were not.
I see. So
definitely, they are not to the extent of what we see in males.
I see. So in male rats, you give ketamine, it causes activity to change in a bunch of different parts of the brain. If you give them ketamine and simultaneously block parts of the opioid system, you see big differences, significant differences in what's going on in the brain. So the opioid system is somehow involved in ketamine's effects. When you do that in females, the difference between the opioid block and non-opioid block condition is much smaller. So there's less of an effect coming through the opioid system in females, it seems.
If at all, yes. There are very minimal effects. We still see a response to ketamine itself.
Mm-hmm.
There are things are slightly different than what we see in males, but still there is a substantial response to ketamine itself. Just it doesn't change when we block the opioid receptors in female rats.
Did you guys go into the study intentionally aiming to look at males and females because there was some reason to believe there might be a sex difference? Or did you stumble onto this in the process of just studying the drug effect itself?
Honestly, we stumbled on this. It was rather surprising that that we saw such a strong sex difference. We only like when we did when we did a preliminary analysis of the data, like an interim analysis of the data that we had collected with about half the animals, then we started seeing these very strong sex differences, and then it became so we had to essentially repeat the studies and to have a better statistical power in both males and females, because then at that point
when you pool them all together, it would have diluted the effect you saw in males.
No, we would still see significant effects even with males alone. So males alone were definitely driving statistically significant effects in the brain. So when we pooled males and females, we would still see effects, but when we specifically used statistical tests to address that question, which is pretty much almost these days it's a routine thing to do. Yeah. Like you use 50% males and 50% females. Just as a caveat, as a historical background, males have been used much more than female animals in research. It's only recently that people have started looking at kind of using both sexes. But anyway, when we do that, at some point we run statistical tests to see if there are sex differences. And so when we did that, we had still a good statistical power. So we had significant effects, but then we saw this very strong difference between males and females. And so at that point, we had to kind of repeat the experiments to validate that and to verify that that was the case, that indeed only males were showing these opioid-mediated effects.
Yeah. So earlier, we were discussing, whether or not, or what the evidence is that ketamine might be addictive to some extent. And we mentioned that, well, it's actually important to think about S versus R-ketamine because there's some evidence to suggest one of them might have more reinforcing properties than the other. Is there the potential for a sex difference here? In other words, does the involvement of the opioid system being stronger in males imply that they might have higher levels or potentially lower levels of addiction liability or reinforcement here?
Again, this is rats, so there is quite a bit in between, between what we show in rats and assessing the abuse liability in humans. Also, I don't think that we can necessarily see that the opioid system is stronger in males. That's not necessarily what we're seeing here.
It's just involved in some way.
It's involved in some way, and it's different between males and females. But I don't think we have evidence to say that at least in our case, that the opioid receptor system is stronger in males. And indeed, I think there is evidence in the literature that it is quite the opposite, that females have a stronger, or have kind of let's say stronger. It's simplifying here, but they have a stronger opioid receptor system. And also in our case, when we looked, now I'm jumping a little bit at the end of the paper with the where we looked at opioid binding of ketamine. Also there, we saw differences between males and females in the nucleus accumbens, but it doesn't necessarily indicate that males have a stronger opioid system. Actually, our evidence seems to be going in the opposite direction, that females are somewhat compensating for that block it than
Yeah.
we do. So we block the opioid receptors, but
Yeah.
females
But
have more
yeah,
capacity.
I guess the fact that you're seeing differences at all here, both the sex difference and with respect to the opioid system, it's probably worth someone out there explicitly testing the level of reinforcing properties you see in males versus females, and then probably breaking that up by S versus R isomer.
Yes, absolutely, absolutely. And yeah, I mean, even the antidepressant effects. So without going in the direction of kind of the reinforcing effects, but even just the antidepressant effects of subanesthetic ketamine, we as relates to the opioid receptor system, we don't know exactly if there are any sex differences there because all the studies that have been done in humans, they are underpowered to, which means they don't have enough subjects to split them in two groups and statistically compare
Mm.
males versus females.
Yep, yep,
So we don't have the statistical power to do those assessments. As to for what relates to the opioid receptor system, not to
yep,
subanesthetic ketamine alone.
yep, yep. So you saw, I mean, ketamine's doing stuff in the brain. It's got this interesting sex-dependent effect here. The opioid system is involved, at least in males. Can you talk a little bit about some of the experiments that you did to look at the nature of how the brain activity changes? You did some ECOG experiments where you're looking at patterns of brain activity.
So the ECOG experiments, they were so as you mentioned earlier, we're looking at blood flows, right? So as a caveat, here we're not looking at neural activity, strictly speaking. We're looking at a proxy for neural activity. Um, so to test or like to address that concern, that actually what we're seeing is indicative of anything happening at the neural level as opposed to just something that happens in the cardiovascular system. We did experiments with electrocorticography. So we implanted electrodes on the surface of the brain, and we recorded electrical signals um, in response to subanesthetic ketamine. And then we correlated those signals with our functional ultrasound imaging responses. But that was with ketamine alone and without the opioid, without blocking the opioid receptor. So it was more a way to validate that what we are measuring here, it is actually correlated to what happens at the neural level.
Mhm.
And what we found was actually very interesting, also because it confirms what other people have seen as well. So functional ultrasound imaging is still a relatively new modality. So we're still, to some extent, trying to really understand the signals that we are measuring, what's the underlying mechanism. But another group have looked at they have looked at kind of electrophysiological, kind of doing a similar study, but in a different part of the brain and with different stimuli. And they saw a correlation between the functional ultrasound imaging signal or the cerebral blood volume signal with gamma power. So when we record these electrical signals from the brain, we split them into frequency bands, and then we can analyze those bands differently, and those bands give us different information.
Mhm.
And so in that case, they observed a very strong correlation between the functional ultrasound signal and gamma activity in, I believe it was the visual cortex and the
Mhm.
hippocampus, and with different stimuli. And in our case, we saw converging evidence that also functional ultrasound imaging was correlated to, very strongly correlated to the electrophysiological signal in the gamma band.
I see. So, and
Um,
when you say gamma band, so basically, there's lots of patterns of like electrical activity in the brain. Um, if by analogy we think about like looking at the surface of the ocean, right? You might have very fast little waves coming, and you might have big slow waves from like a big ship going by. You guys can sort of measure how much of all of the different wavelengths there are in the signal, and you're saying that you see certain changes in certain bands. So the gamma band would be relatively fast rhythms in the brain, right?
that's correct. Yes, that's in our case, it was between 30 and 80 hertz. Everyone uses different kind of limit for those bands, but it's usually between 20 and 30 and 50 to 80
Hmm.
hertz, the upper band.
I know that there's, a lot of people have a lot of different opinions in the neuroscience world about what all of these rhythms and frequency bands mean. But just to give some people a sense for something a little bit more concrete here, what would be like a naturalistic behavior? Like, do you see high gamma band activity in awake animals, in sleeping animals, in animals that are like doing a task, in animals that are eating food? How do we think about high gamma band activity?
Well, I mean, that's not really my field of study, so I kind of, yeah, that's just, a big caveat there. But high gamma is mostly kind of indicative of kind of an awake and aroused response, so to
I see.
speak. Yeah.
Okay. Yeah, and I guess that would make sense. I mean, the doses of ketamine you're using here are, by design, not put they're not anesthetic.
Exactly,
Yeah,
exactly.
yeah.
So that's expected. Um, yeah, and also interestingly, there is, and again, this is a kind of, it will be object of further studies because I think it's something very exciting that kind of the interneurons where ketamine binds at the NMDA receptors that we mentioned earlier is one of the hypotheses of subanesthetic ketamine. They're also involved with generating gamma oscillations in that part of the brain. So I think that was a very interesting kind of point of contact between the two, which I thought was very, very exciting. But we don't have evidence to show that those are related. But I just thought it was very interesting that we did see those patterns converging between the two modalities in that part of the brain, which was the prefrontal cortex again.
Mhm. Um, and based on this study or other studies that are out there, when you give ketamine to a rodent, a freely behaving rodent, what are the behavioral effects? What are they? A sub-anesthetic antidepressant dose. What do they do? How does their movement and their behavior change?
Um, I mean, there are different patterns, at least in rats. Um, they definitely become wobbly a little bit. I mean, they show kind of their behavior changes, their patterns of behavior change in a way that they are kind of less in control, like so to speak, of their environment. I'm just anthropomorphizing the rat here, but they seem to be kind of less aware of what's happening around in their environment. There is a hyperlocomotion response, so they are actually
Just moving around more.
moving around more, which is actually something that we also tested, and that is also something that has been shown quite a bit in the literature that ketamine has causes hyperlocomotion in rodents. So they're moving more, but kind of less able to move overall. They also sometimes show stereotypical responses, like they are maybe grooming more, or they can, yeah, do kind of different stereotypical
Mhm.
behaviors, but it's usually short lasted. So within a few minutes, they are pretty much back to normal. So it is
I
usually
would see.
very, very quick. Yeah.
I see. Yeah, I would. Um, yeah, and ketamine doesn't last that long. Like in humans, right? It lasts for a shorter period of time than something like psilocybin, say. And because of the metabolic differences between rodents and humans, I imagine that the drug is basically in their system for just minutes, right?
Yeah, it's very short. I mean, just also another thing that we should mention is that the dosage is very different
Mm.
between um, humans and rodents. Um, And that's again because of the differences at the metabolic level. The way the drug is metabolized in the two animals is very different, and rodents have a much faster metabolism. So usually we give the doses that we give in animals are much higher than what is actually used in humans.
Hmm.
But those are kind of the doses that have been shown to have antidepressant-like effects in rodents. So there is a lot of literature showing that at that specific dose, um, you have antidepressant-like effects, which is 10 milligrams per kilo of body weight.
Mhm. So what are you guys doing to follow up on this, or more broadly speaking, what do you think are some of the next steps for the field to get a better handle on ketamine's mechanism of action in the brain?
Um, well, I think at the rodent level, one thing that we are actively investigating right now is trying to understand better that circuit. So now we got these snapshots of regions in the brain where something interesting is happening when we block the opioid receptors, but we don't know exactly how they relate to each other, and we don't know exactly what is happening when these regions, how these regions talk to each other, essentially, and how those patterns change with and without the opioid receptors. There are some other regions in the brain that are definitely interesting to look at that we left out in our study because also we had to kind of decide because of the setup that we had at the time. So now again, since we have access to kind of volumetric imaging, we're also expanding that. So there's a paper, it's actually a preprint that just came out a few weeks ago where they also show interesting effects in the central amygdala, also with opioids and ketamine. So that's also another region that it could definitely be interesting to look at. But in general, I mean, I'm interested in having this whole brain picture of what's going on with and without opioid receptors. But that's all in rodents. Um, And then in humans, it would be, I think it would be very nice to see if these kind of the effects that we found or this kind of ketamine opioid interaction and the sex differences translate into humans. So we don't have a whole lot of fMRI literature with ketamine, so it's something that there's definitely some, but it's not a lot. So we don't know exactly how those patterns may change in humans, but that I think would also be very interesting. Try to understand. Essentially, the question is if opioid receptors are a thing at all, and if there are those strong sex differences, we need to try to understand if that is the case before deciding what to do with it, essentially, before kind of steering the field in one direction or the other. But I don't think we have a clear picture yet of what's happening.
On the one hand, obviously we have seen that ketamine can have these remarkable rapid antidepressant effects. They don't last forever. They are short term. Um, on the other hand, there's still a lot of open questions, right? There's still a lot of controversy and work to be done in the field to work out things like sex-dependent effects, involvement of the opioid system, and the full mechanism of action to be worked out, and what exactly the addiction liability is. Is it different between the S and R isomer? Is it different between males and females? So we've got all of the science that's still ongoing in terms of how ketamine works and what it's doing. And on the other hand, because of the remarkable results with respect to the rapid antidepressant effects, we've seen things like ketamine clinics just pop up all over the place. I see them all over now. To what extent, is that warranted? Because we're seeing these rapid antidepressant effects and they are apparently helping a lot of people. People are commonly reporting that it does in fact help them when they get these doses. And to what extent, should we maybe be a little bit more cautious because while we are seeing these rapid antidepressant effects, we also know they don't last forever, and there may be things we don't understand that should make us slow down a little bit.
Yes. So on that note, I wanted to mention before answering your question that since ketamine has to be essentially administered over and over again, we also looked at repeated administration, effects of repeated administration of ketamine. Um, because we kind of the rationale there was that we did see opioid receptors modulating the response to ketamine in that reward system. And also knowing that ketamine needs to be administered in humans repeatedly, we wanted to try to see if the opioid receptors were also having an effect on kind of in that specific
Yeah.
behavioral response.
Like if there was a
Um,
habituation to the response or something
exactly,
like that.
exactly. And so we looked at locomotor sensitization. So as I mentioned, rats and mice, they they show this hyper locomotion when they are administered ketamine, meaning that they behave more, they walk around more acutely. So in the first 20 or 25 minutes of ketamine administration, after ketamine administration. And as we administer the drug repeatedly, that response goes up. So the animals show some sort of adaptation in that part of the limbic system that is ramping up that behavior over several days of repeated drug administration. So we also try to probe that, and again we saw that naltrexone blocked that behavioral sensitization, locomotor sensitization in males. And in females, we still had a behavioral sensitization, meaning that the animals were behaving more and more, like they had a locomotor. This locomotor response was increasing more and more over. I don't remember if it was. I don't recall if it was 4 or 5 days of repeated administration, and but we could not block that with naltrexone. So when we blocked the opioid
Mm.
receptors in males, we were essentially deleting almost completely that behavioral sensitization, while in females that was not the case.
I see. So again, there's a sex-dependent involvement of the opioid system here. What exactly is going on, we can't say. But behaviorally, the males were becoming more sensitized to the effects of ketamine, at least with respect to locomotor behavior.
No, they were both becoming sensitized to ketamine, both males and females.
I see,
But in males, we could block that effect by blocking the opioid receptors,
but not in
but
females.
not in females. So in females, whether the opioid receptors were available or not, they would become more sensitized to
I see.
ketamine regardless. But that was not
But
the case in males.
got it. And so, what would this potentially imply? That potentially you could take lower and lower doses of ketamine and you would still get the same magnitude of effect? How should we start to think about this?
Um, I think it's more of a safety question. I mean, I think taking a lower and lower dose of ketamine, that would be more
Well, let me
a
restate
question
that.
of efficacy, like
Yes.
whether it
So,
is, whether,
yeah.
sorry, go ahead.
So if there's sensitization, and let's say someone's going into a ketamine clinic and they have to go back every 2 weeks because the antidepressant effects aren't permanent, would this imply the possibility that if they're going back once every 2 weeks, they're actually becoming they're effectively getting a larger dose because they're becoming more and more sensitive to the dose they're getting?
Um, Not necessarily. I mean that because there is also like a tolerance effect, which means they are kind of the opioid receptors specifically, they are changing in response to repeated administration, and that's actually something that we showed both in this paper, but also in the other paper that I mentioned with S-ketamine. We show that the repeated administration of S-ketamine is reducing opioid receptor availability in the nucleus accumbens.
Mm.
So basically, when you lower the level of the opioid receptors available, essentially you need more drug to get the same
The same
effect.
level of reward.
Exactly. So
Yeah.
it's not necessarily um, it's not necessarily like that they're getting kind of a higher effect.
Mhm. I
The
see.
locomotor
Okay.
response is not, yeah, it's not necessarily indicating that.
I see. But, with repeated dosing, there are differences. So it's not the exact same effect you're seeing with each successive dose.
Exactly. There is a sensitization and there is a tolerance effect
Mhm.
mechanism that are going on when you administer the drug repeatedly.
Mhm. So that would imply the likelihood that if repeated doses of ketamine are given, time after time, the way the person or the animal responds is going to change over time potentially.
Yeah, that's correct. And it might, like in the case of again, like with S-ketamine, what we were probing there, and those were experiments that were done at NIDA. Again, we did the functional ultrasound imaging experiments for that paper. That is again like showing that the animal is developing some sort of tolerance to the drug. So it's reducing the sensitivity to the drug because it has less opioid receptors to be activated.
So what are some of the experiments you guys think you're going to do in the near future? What are some of the next steps and the next questions?
Well, as I mentioned, for us, we're very interested in trying to kind of go deeper in understanding the circuit level effects, trying to understand how those regions talk to each other, and how that changes in response to that opioid modulation. That is definitely something that we are actively doing right now in the lab. There are also like another aspect of this. So the lab where I was before, where I did this work at Stanford, is developing technologies to deliver ketamine in a focal way. So kind of deliver ketamine only to specific regions in the brain, also using ultrasound. And therefore, another aspect of this research is trying to understand if there are regions in the brain where we can preferentially elicit, let's say, now I'm stretching a little bit here, but let's say that we can preferentially elicit an antidepressant response versus other adverse effects. And therefore, if that is the case, we can deliver ketamine only to that region specifically.
I see. Yeah. So in principle, the antidepressant effects might come largely from ketamine's effect on, one part of the brain or one circuit. Maybe other effects come from other places. If you could locally apply it there, the idea would be you get the antidepressant effects without the other stuff that might be undesirable.
That's correct. And there have, like in rodents, people have, for example, delivered ketamine, like that you can microinfuse the drug in specific regions in the brain. So people have shown definitely that ketamine in the prefrontal cortex can have an antidepressant effect. People have shown that ketamine in the lateral habenula, this very tiny region where we also saw opioid-dependent responses. So infusing ketamine only in that region also seems to be sufficient to elicit an antidepressant effect. So there are indications that actually that may be sufficient, kind of that you can target regions individually and still have an antidepressant effect. Now, we don't know if that antidepressant effect is, again, mediated by the opioid receptors in that region, and if that would be necessarily safer. That's an open question, but that is another aspect of this kind of mapping that we're trying to do here.
Interesting. Well, this is exciting stuff. Obviously, this is a pretty hot field. There's a lot going on, and it's pretty fast moving. So it's exciting. Is there anything you want to reiterate for people, or any final thoughts you want to leave them with about the specific work that you did, or just this field in general?
Just again, I think that we are, like, as basic scientists, we are putting out evidence, right, that something is happening, then at least in rodents. So I think these are definitely effects that need to be confirmed in humans. So hopefully there will be clinical trials to specifically test these questions, because I think it is an important, it's critical to try to understand better these mechanisms to improve the safety profile of ketamine. And if we have sex differences that have not been tested specifically so far, it will be critical to try to understand those better and try to understand if that is actually a real in humans. So,
All right. Well, Professor Tommaso Di Ianni, thank you for your time.
Thank you so much for having me.
Hey everyone, I want to take a minute to tell you about a really cool health monitoring device I've been using for several weeks. It's called Lumen, and it's a handheld, pocket-sized device with a sleek design. It measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs. It improves your ability to burn fat, which decreases your hunger levels and makes your body less dependent on snacking, and it can increase your energy levels by helping you develop a high-functioning metabolism. I use this device in the morning, before bed, and after meals and workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I need to fast each day to promote fat burning. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. The easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MIND, M-I-N-D, in all capital letters to get $50 off your Lumen device today.